Biology Part 197. In a mature anther, the diapetrum shrinks so that the pollen grains are suspended in a hollow sac, while the outer sterile cells become fibrous. Strips of uneven cellulose thickening are laid down in the epidermal cells at each side of anther in preparation of the dehiscence, or splitting, to release the pollen grains. The outer surface of the pollen grain is often beautifully sculptured with pits and projection of the forming of an intricate pattern which is characteristic of each individual species of flowering plant. In addition, the material from which the exine is made, called the sapropollenin, is extremely resistant for allowing the pollen grains embedded in the sediments or peat to remain unchanged in the appearance for thousands of millions of years. The science of plant identification from pollen grains is used to determine which species were present in the vegetation at the particular times in the past, and gives an indication of the likely climate even in the absence of other plant remains. Embryo sac and ovum. The process of leading to the formation of a female gamete or ovum is summarized in our figure. As the carpel develops in the flower, a small bulge of nucleus grows out of the region of tissue known as placenta. The nucleus consists of a group of unspecialized nutrient-rich cells which divide meiotically to form an oval mass. As it swells, a pair of covering integuments grows around the outside, eventually leaving a single tiny opening. And the micropyle, the whole structure, is called an ovule and is lifted clear of the placenta by a stalk or funicle. During this process, a single large cell called the megaspore mother cell begins to divide by the meiosis inside the nucleus. This division results in 4 megaspores, but only one survives. The other 3 are broken down and absorbed back into the nucleus. The single remaining megaspores enlarges and its nucleus divides mitotically 3 more times to produce 8 haploid nuclei inside a structure which is now called the embryo sac. In terms of its development, this is equivalent to the pollen grain of the male. An important difference is that some of the nuclei within the embryo sac are closed within membrane and cytoplasm from the separate cells. The ovum is one of 3 such cells situated at the micropyle end of the embryo sac. It is flanked by 2 other cells called synergids, forming an arrangement known as egg apparatus. Of the remaining 5 nuclei, they are located in the antipodal cells. At the opposite end of the embryo sac, the 2 called as the polar nuclei lie close together near the center of the embryo sac. The polar nuclei are not normally endorsed by the membranes and usually fuse to form a single diploid nucleus. Thus, a mature embryo sac contains a total of 7 nuclei comprising of the ovum, 2 synergids, and 3 antipodal cells in addition to the central diploid nucleus. Pollination mechanisms. The transfer of pollen grains from anther to a receptive stigma is called pollination. In self-pollination, this transfer takes place within a single flower or between 2 flowers on the same individual. In cross-pollination, the flowers are located on different individuals Cross-pollination tends to result in a greater variability among the offsprings and is often favored by natural selection. Consequently, many species of the flowering plants possess adaptation which make cross-pollination more likely.